So what I'll talk about today is research that started during my PhD time in Gothenburg under first Peter Mostad and then Serek Sakitov, and that I've come back to in a couple of years ago. So it'll be a mix of some statistical inference and some probability. And I'll first start with an introduction to what I am working on, and then I'll talk a little bit about go through a worked example that talks, that gives the biological motivation for all of this, and then end, if time permits, with some more probabilistic mathematical results. So what are phylogenetic comparative methods? So if one goes into nature and starts observing species that are closely related to each other, one will notice a diversity of traits in them. So for example, here we have Southern African bishops and weaver birds, and we can notice that some of these bird species have got very, very long elongated tails. Others have got very, very brightly colored plumages of these males. Okay, and we would want, or biologists would like to study these traits. For example, they could be asking questions whether there is a relationship between whether a species chooses to have long tails or brightly colored feathers with relationship to some environmental factor. In fact, in this particular case, these long tails, they are in species that tend to live in open savanna regions, while the brightly colored plumages are there in regions which are more densely bushy, bushy regions. Okay, and so of course this can be quite easily seen. But now, if we want to study these things more formally statistically, so we have a statistical sample. We have measurements of these tail lengths and colored plumages from a bunch of species that are captured and measured. So if one wants to make a statistical analysis on the between species level, one cannot forget that these are not independent measurements, that these data points are dependent through the shared evolutionary history of these species. And once one has a dependent sample, one needs to introduce some stochastic, some mathematical framework for modeling and dealing with these dependencies. So the framework that we are dealing with here is that, well, here one has a phylogenetic tree joining the species today. So this tree, this graph, represents evolutionary history of, say, the species that are the tips here. So in this example, we have 4 present-day contemporary species. Okay, and they evolved back in the past. So the horizontal axis is the time axis, sorry, the vertical axis is the time axis, and with time running up, well, downwards. So the past is here at the top. Okay, and we can see these 2 species, they diverged, or they had the most recent common ancestor here. Then this clade of 3 species had the most recent common ancestor here, and all 4 of these species had the most recent common ancestor here. Okay, right, so this is the evolutionary history that is, well, somehow estimated, usually through genetic data. It's also somehow time calibrated. And then on top of this tree, we have a stochastic process that evolves. Right, so here we have a Brownian motion evolving on top of this tree. So we have, say, the blue lineage that comes onto this point here, and this point, it diverges into 2 separate lineages, and also the red one continues along this line here. Then we have then we reach this point here where the orange lineage diverging off, and until we reach this point here when you get the split into the green and the blue. Okay, now if we can imagine that the x-axis, or the axis of trait values, is the horizontal one, right? Then we would have that the values of the traits would be on this one, on this horizontal axis, with the time axis being the vertical one. And then, well, what we actually measure only today are the values of the traits at this point here, 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 and here. We don't measure this whole trajectory that all this family of trajectories that have led up to what we have today. We can only really measure that what the species that are alive today. So we only have these 4 data points. We have the dependency between these data points induced by the tree, and we have some stochastic model for the scalar Brownian motion for the traits evolving on this tree. And the aim of the statistical inference is then to estimate the parameters of the stochastic process while correcting for the fact that our sample is dependent. So I gave the example as a Brownian motion, but then of course that by itself would not be that interesting. We need to have some family or some framework of stochastic processes, and with Vanillin Mitov, who did his PhD under Tanja Stadler at ETH Zurich, we developed a framework which is called the GLInv family models. So it's the Gaussian and linear and invariant variance family models. I'll explain what is really meant here. So Vanillin first developed this computational statistical approach for Ors and Unweg processes, and after we met, we discussed and noticed that this can be extended to a very, very wide family of Gaussian models, and one can also go potentially beyond the Gaussian family. So there are 2 main assumptions. Well, the first assumption is a Markovian assumption that after branching, the traits evolve independently in all descending lineages. So after this branching point, the blue lineage evolves completely independently of the red lineage. Here, after the branching, the orange lineage is completely independent of the blue one. They can cross, but these crossings are completely meaningless. Okay, so we have this sort of Markovian property. And then the second distributional assumption is that the trait, if we denote by X the trait process, so conditional on the past, it is Gaussian, and it has the following properties: that the conditional expectation is linear, based conditional on the past value, but more importantly, that the conditional variance is completely cannot depend on the past value here. So these parameters omega, phi, and v may depend on time here on these at the time points here. So they could vary over the tree, but they have to be independent of the trait value itself. Okay, and what can you do with this? Well, phylogenetic comparative methods, or say Gaussian processes on this sort of trees, they have been around much earlier than my joint work with Vanillin. But the main problem was that if you want to inference, you need to get the likelihood function. And if I have a multivariate normal sample, so notice here that because this is a dependent sample, I have these 4 measurements here, and if we assume Gaussianity, then these four observations, they will be just they'll come from a 4-dimensional normal distribution. Of course, we can have many more species. So we could have maybe not 4-dimensional, but 60-dimensional, 100-dimensional, 1,000-dimensional, 10,000-dimensional normal distribution with a non-sparse covariance matrix between them. Okay. Right? So we can, of course, write out the likelihood, but then we run into computational bottlenecks that actually constructing V is done in K-squared time, where K is all the observed traits in all tip species. So we have n tips. Each species can be described by a vector of traits, which could be of the same dimension for each species, but some species could have some traits missing, non-observed, or non-existent. So in principle, we can have a different dimensions for each species. So, okay, so we have a K by K covariance matrix that takes at least K squared time. Okay, and it will take K squared time to construct, and to calculate its inverse and determinant will take us at least k to 2.373 times, depending on the algorithm which is implemented, which means that these sort of things can be done for a few traits. So if we just stuck with this method, from my experience, you could do maybe 6 or 7 traits and maybe 300 species. Maybe you could, with fewer traits, you could try to push this to maybe 1,000 species. But in general, this was not sufficient for any modern applications where you have phylogenies with a couple of hundred, couple of thousand, or even up tens of thousands of species. So with vanillin, we're discussing, and what we discussed was that, okay, well, first of all, if we knew the values of the traits, if we had fossil measurements, or we could go back in the past and measure the trait values at the internal nodes, and there's a lot of notation here, so maybe some of this I will explain, some of this is not that relevant, but I'll first try to go over the main ideas here of this picture. So of course we have here a phylogeny with 5 species. We have here, and we have the internal nodes in this tree, and of course if we knew the internal node values, internal nodes in this tree, then to get a likelihood would be straightforward. One would just take the product over the likelihood of the transition between each of these nodes. Of course, we don't know the past, but if we could integrate in some smart way over the internal nodes, then we could get an analytical formula for the likelihood. And well, what we noticed then was, okay, well, we can't have a nice closed form formula while integrating out the values of internal nodes, but what we can do is something else. So there's again a lot of notation here, but this is if one looks at this on a high level, it's pretty straightforward. So this is just another way of writing the likelihood of a multivariate normal distribution, which will be some sort of a quadratic form in terms of the, this is our current measurement conditional on a past measurement, so this becomes some sort of second order matrix vector formula. And then the key thing is that these A, B, C, D, E, and F terms, of this representation of the Gaussian density, in our case, will be in terms of the covariance matrix change along a particular lineage, and then in terms of this omega and phi that represent the conditional expectation here, right? So basically, I'm rewriting that quadratic form in terms of these omega, phi, and v. And then again, it's a lot of notation, but the key thing that one can do when one would analytically integrate out the internal node values is that one gets that the likelihood of all the measurements over the tree can be expressed as this sort of quadratic form with these matrices and vectors. Well, they can recursively represented in terms of, well, these parameters or these matrices that are functions of our underlying model, and they are particular to each branch of this tree. Okay, so we have a recursion over the tree. Okay, and the thing is that we need to traverse each branch only once. So now, instead of using up a quadratic amount of time in the number of species to create the likelihood function, then to take the inverse of this quadratic sized covariance matrix, we can in linear time traverse the whole tree and get the value of the likelihood function that is conditional on the root of the origin of the tree. And then one can do maximum likelihood, for example, maximum likelihood for this origin that's conditional on the tree value, well, and the set of parameters that are these omega, phi, and v matrices and vectors that can be branch specific. So if I go back to this picture, the important thing is that we are integrating out these things. We are going lineage, we're going branch by branch, okay? And now we can have parameters that are branch specific. So it means that we can have for different parts of the tree, phylogenetic tree, which means that for different sets of species, we can have different parameters which will hopefully correspond to different biological hypotheses. We can have an arbitrary collection of missing values for each tip species, right? So notice in this picture I have NA, so missing values for, say, the second and fourth coordinate, and so unfortunately very typical in these sort of studies, well, that it's for some species it's not possible to collect all measurements. We can also assume that, and this is there's some different mathematical treatment, that some tips that some traits are missing for some species. For example, the simplest example is that humans don't have tails. So for some primates, we have the tail length as not as a missing observation, but just a non-existent observation, right? So there is some slight mathematical or different mathematical treatment. We can have in particular that all the tip branches don't need to be at the same height. So on the previous example, I said, okay, we have measured species that are contemporary. So the town calibrated phylogeny, these species should be the same height, but one can imagine that these are fossil species, or one does not have a time calibrated phylogeny, but it's measured in time mutation or numbers of mutations. So it's a non-ultrametric tree, so there's no reason for these tips to be the same height. So from the perspective of this, sort of framework, it's completely irrelevant. All of these things are irrelevant, which were very important in previous previous implementations that are more biologically motivated, that you could have methods that worked only for ultrametric trees that would not allow for missing values, and so on and so on. So in this general framework, we can essentially have any Gaussian process that goes on top of the tree. And so we implemented this in a family of R packages. So with the so-called PCM base package, we provided a user interface that a user can specify any collection of models that evolve in the phylogeny, and then this PCM-based package just returns the likelihood function, and then the PCM-based fit package, which still is not on CRAN, it's on GitHub, that actually does parameter estimation for these packages. We decided to split the estimation and the likelihood calculations separately because we would want others to be able to use these packages, that they can implement their own model, right? They don't only have to use what we have the time to provide, right? And importantly, this PCM-based, the likelihood calculation package, has a C++ version, which speeds up the likelihood calculations, well, something like 10 or made up to even 100 times, but on average is about 20. And then during my PhD time, I developed the so-called MV slouch package, which is multivariate or Ornstein-Uhlenbeck models for comparative hypothesis. So this was developed in a multivariate OU process on top of this tree, but then with the collaboration with Vanillin, with using this PCM-based package as a computational engine, it speeded up the calculations about 50 times, or even more. No, a couple of 100 times. I would say that running some experiments, I was able to do estimation for a tree with 250,000 tips in 3 days. So to get an idea, previously, so without this sort of linear time likelihood calculations, it was possible to get within 2 or 3 months estimates for about 300 tips, right? So we can see that this is it opens up very new possibilities. Okay, so to give an example of things that we like to work with, I will talk about the evolution of the fruit in the genus Ferula. This is data that is provided by Dr. Marcin Piwczyński from University of Toruń. Then I'm collaborating with him on developing of applying these fast inference, phylogenetic inference methods for, say, or in this case, the Ferula, but also these Ferula fruit are giving us very nice feedback in what direction we should do the method development. Anyway, Dr. Piwczyński has a data set of 78 Ferula plants that can be monocarpic or polycarpic, meaning that they can produce seeds over many times, or they just produce seeds once in a lifetime. And he has measured a number of properties of these fruits, well, their wing area, some information on the canals inside these fruits, how thick they are, and the maximum height of the tree and the mass. So from our perspective, we really have we have 1, 2, 3, 4, 5, 6 continuous variables that jointly evolve on this tree, and we have a binary variable that is a certain one level for some of these plants, another level for different of the splits. Okay, and we want to essentially study how these traits co-adapt, whether any irregularities can be seen, whether we can see which traits influence which traits. And of course, we need to have very specific models, and we need to have interpretations of the model parameters that are related to this set of traits. So we consider the Ornstein-Uhlenbeck framework here. So the simplest one is a Brownian motion, when basically we have no stationarity in the process, or we have the Ornstein-Uhlenbeck process, where if this drift matrix has got a positive real part eigenvalues, then we have convergence to stationarity of the process. And we have the so-called mixed Ornstein-Uhlenbeck Brownian motion. Of course, this is of course an Ornstein-Uhlenbeck process with just zeros in appropriate places on the drift matrix, but for computational reasons and for interpretational reasons, we want to split the coordinates into two sets. One is the x's that just follow a Brownian motion, and then the y's that conditional on the x's, they would be following an Ornstein, or they would be kind of following Ornstein-Uhlenbeck process in this formulation. Okay, and we have adaptive hypothesis. So first of all, we want to see whether whether a pure Brownian motion would fit better, whether an OU process of this sort would fit better, or whether a mixed process with some of these traits evolving according to a Brownian motion would fit better. Then the other question that we want to ask is, can we actually go one step more and formulate hypothesis on the structure of A? So for example, we want to ask, does the data support a diagonal A better, or a triangular, or maybe a symmetric positive definite A, or maybe a general a non-sparse A that also has, that we assume that has positive real part eigenvalues, so that we have convergence to stationarity. So we want to distinguish between these sorts of hypotheses. Okay, and so in order to do this, we need to set up these sort of things in terms of biological terms. So we have two. So Dr. Piwczyński suggested two classes of models. One where you have this live strategy, the live strategy binary variable present, and he asks, okay, whether does live strategy influence max height and the maximum height and seed mass. And the way that this this influence is done is that this sort of stationary expectation of the OU process is different for each live strategy. Okay, and the second class of models are the relationships between these 5 continuous models that we had. So we want to formulate whether or some, or whether, for example, seed mass influences only these 2 variables or these 3 or 4, and whether there's a influence between these 2 variables in evolution. And the way this is done, I'll show on the next slide, because next slide is more interesting, that it will correspond, or biologically it's interpreted as corresponding to zeros in appropriate places in this drift matrix. So the model that was found was that in this old study, so actually when we updated the study to with well, we're running further and further analysis, so we had we actually found that a completely different model fits best, but I'm discussing the results from some previous estimation runs, because I just think they're more interesting to discuss and present here, while under the caveat that further analysis actually pointed to a completely different model that was not that I don't want to discuss, because it's a much simplified model and it doesn't it would not show what we could actually do, how we are modeling this. So how, as I said, how would we the first set of models were the last strategy influencing maximum height and seed mass. So again, this corresponds to different values of this drift matrix for polycarpic and monocarpic levels. Okay, and we found this set of estimates fitted best. So we had a drift, or some sort of drift matrix, and this sort of drift vector here. So under the model where this drift vector was separate for monocarpic and polycarpic plants, you can see that these values were very, very close to each other. And this is something that is actually often observed, that a biologist will come with a hypothesis that these optimal values, or the stationary mean values, should be different for many different levels of some discrete variable. However, the software points very clearly that some of these levels can be lumped by just indicating that they look very, very similar to each other, the estimates here. Okay, and well, we can also get confidence intervals for these values here. Okay, and the confidence intervals for the drift and diffusion matrices, they are a bit more difficult to get. While these values we could do through some regression, it's, well, the only way of getting confidence intervals at the moment for the other parameters is through bootstrapping, but hopefully this will change in the nearby future. Okay, then as I said, the more interesting thing is when we want to study these other 5 traits, and we want to say formulate hypothesis that these 2 traits influence each other and don't influence any other, while the seed mass has an influence only on these 2 traits. So the way one does it is one really when we have variables which we say, okay, there's no influence between them, we put zeros on the drift matrix, while if we have one-way influence, we put non-zero values in one direction here, while we assume 0 values in the other direction. Okay, so we can formulate a lot of hypotheses in this way. We're just setting 0 or non-zero values. Non-zero meaning that we want the program to estimate these values, while positive pluses mean that we want to force these values to be positive, and negative minus would be that we're forcing this value to be negative. Right, so we can formulate it in different ways. So we have this one that the ratio canals is not influenced by anything, but seed mass influences 3. So notice that we change these values, force these values to 0, and put a question mark here, or we can say the seed mass forces everything here. Okay, so, and in the first initial study that we ran that pointed to that the best model was the one which is here, that seed mass influences everything, while seed mass itself follows a Brownian motion. So by best we mean means that best means according to archaic information criteria corrected for small sample size. So this was done a couple of years ago, and then more recently we again looked at this data. What we found was that the best supported model was one where this A is diagonal. These values are set to 0. However, this diffusion matrix is such that these values are non-zero, right? So basically, in one way, then after a more careful study, it was that these non-zero values are coming from here to here. Anyway, but I'm showing this all the results that are improved later on because I think it's just more interesting to show that you can have these sort of hypotheses. Okay, and what is interesting in the model where some variable is assumed to behave as a Brownian motion is that these parameters here, so this the way it influences, they enter the conditional expectation for these other variables in a linear fashion, so one can have a iterated GLS actually to estimate these things. So in principle, an iterated GLS should be more efficient than just numerical optimization of all the parameters, but we got some interesting estimation issues that we noticed in the sort of set that if we take these B's, so these the ways that the parameters that mediate how the Brownian motion parameters, Brownian motion variables influence the non-pure Brownian motion variables, it seems that the iterated GLS has some sort of asymptotic bias towards it. So this is the estimation error of this whole vector of parameters. So this is absolute here in the first column and relative estimation error in the second column. Okay, so if we do this sort of iterated GLS, which I'll just present in the slides, in the previous slides, we get some sort of asymptotic bias. Well, if we just do numerical maximum likelihood over these parameters, this at least there are indications that of this numerical bias might not be present. So these are errors on the logarithmic scale. So you can see that this is e to the power of -2 - 3. So that's a relative error for 4 parameters together. Okay, so again, so the more interesting statistical thing is that sort of the estimation issues that come up when one starts running these sort of numerical likelihood optimization methods. Okay, so the first thing is how should one seed these unknown OU matrix parameters? So one can of course calculate the basic seeding idea is that one goes and does some sort of rough one-dimensional analysis for each trait, and one tries to plug in these things. One basically takes the sample mean and the sample covariance, and based on the sample mean and sample covariance that ignore the phylogeny, one tries to preset the parameters of this OU process. It was interesting enough that up to now we did not really notice any significant improvement. One just has random starting parameters or ones that come from estimation when one ignores the phylogeny. But I said more interesting is asymptotic bias for this parameter of the interaction between the Brownian motion and non-Brownian motion traits. So one could show that conditional on, say, the drift matrix and the diffusion matrix of this OU process, these B's, they enter the conditional expectation of the non-Brownian motion traits linearly. So one would start an iterated generalized least squares. That was done because in the previous, say, slower versions of MVV-Slouch, it was impossible to optimize all parameters of the drift matrix by numerical maximal likelihood. So we developed an iterated GLS. So basically how it works, well, we condition on the drift and diffusion matrices. Conditional on these, you do a generalized least squares to get this B parameter, and then you, in conditional on this B parameter, you optimize over A and over the drift and diffusion. Okay, and now once with the new faster linear likelihood methods, we're able to just basically do nothing interesting and have this whole drift matrix, including these B's, to be estimated by numerical maximal likelihood. And there is a significant improvement, though it's an open and I think interesting statistical question. Why is there this asymptotic bias in these general least squares methods? Is this due to maybe some sort of error in variable formulation, or whether there is something more interesting going on? So because in principle, I mean, the results, I mean, the theory would tells us that these two sort of things should converge for large enough data, in the independent data case, to the same estimate. But it's interesting to study now whether this, what does the phylogeny bring into these generalized least squares methods that cause problems. Okay, so I think I have 5 minutes, so until 2 o'clock. So I will just go quickly through some asymptotic results that we also worked on. So previously this was some statistical estimation approaches, while we also worked on some probabilistic properties of these branching Orsa and Uhlenbeck processes. And the key thing here that we are now specializing, and we assume that the phylogeny is modeled by a pure birth tree. So it's there's no extinction in it. And the key property of these trees is that between speciation events, the length of these between speciation times are exponential, with rate equal to the number of lineages times some speciation, right? So just to say one thing, of course, these between speciation times are drawn not to scale. I mean, they should become shorter as time progresses, because this will be exponential 1, exponential 2, exponential 3, exponential 4, exponential 5. So they have shorter expected values, but then if these would become shorter, this picture would be completely unreadable. So, okay, so the first set of things that we showed during my PhD time with Mitov was well, depending on the OU process, if we look at the sample average and the sample variance, well, then we can show consistent, well, as long as it's also an Uhlenbeck process, we can show that we have a consistent estimation of the stationary variance of the OU process. We have consistent estimation of the stationary variance parameter of the Orsa-Uhlenbeck process. We have we can't have inconsistent estimation for the Brownian motion process of the ancestral trait, or the origin of the tree, which was also something that was well known in the literature before. But what was also interesting that we managed to show is that you have a consistent estimation based on the sample average, or the sample variance of the diffusion parameter of the Brownian motion process. And then we could also get the variance of the sample variance. So while it is pretty straightforward to show that the variance of the sample variance tends to a constant, well, after logarithmics, it just tends to a constant, then to show exactly what this constant is actually costs quite a bit of effort. And beyond that, we showed central limit theorems, which then I have worked on in the past couple of years with Torkel Erhardsson from Linköping University. That so with the SERIC we showed that you have convergence in distribution to a perturbed standard to a mean zero normal distribution of the centered and scaled sample average. Then with Torkel here in Linköping a couple of years ago, we showed rates of this convergence. So in the Kolmogorov distance, we can show that we have this sort of rate of convergence as the number of tips increase. So as the pure birth tree grows, we can show this rate of convergence. Also, we showed in the Wasserstein distance, but I'm not showing it because these rates of convergence or just take the constants would just take the whole slide. Okay, so in these sort of central limit theorems, we actually have three classes. So this is some sort of phase transition. So when the drift, the coefficient of the OU process is greater than half the speciation rate, then we get just something that looks as if we had a usual central limit theorem. When it is at equaling the speciation rate, we have still something that looks like a normal central limit theorem, except that the scaling is a bit different. Here we still have a logarithmic term, and we can also show convergence rates of this convergence in the Wasserstein and Kolmogorov distance. But when we have slow convergence, or that the drift parameter is lesser than half the speciation rate, we can still show there is convergence, which is almost sure L2 to something, but we can't say anything about the distribution of the something. We can just say, okay, that this is some random variable that has finite mean and finite variance. And finally, what we also generalized these the sort of Uhlenbeck processes, we introduced models with jumps at speciation points. So this is kind of biologically interesting that when two species diverge, that they form different species, that may be some sort of very rapid or very quick changes in the traits took place, which would mathematically correspond to just a jump taking place. So here we have our trait that's evolving on the phylogeny. Here's a speciation point, and at the speciation point, this trait just had a jump in the value along one lineage. Okay, so if we assume it's a mean zero Gaussian jump, then we can show this same sort of central limit theorem. Okay, basically, that says that if we have this jump is something, then and we have fast adaptation, then we can show that the characteristic function of the scaled and centered sample average goes to the Laplace transform of something, which is it's an ugly central limit theorem that maybe we will be able to improve. Okay, something like this happens also. So more interestingly, when you have the drift parameter is equal to half the speciation rate, we can explicitly show that there is convergence to some normal distribution with rates of convergence shown. Well, and again, when you have slow convergence or when you have slow evolution, then not much can be shown. But however, interestingly enough, that if this so this was when this jump probability, so the probability of having a jump is constant. However, if these probabilities, or rather the probability of the jump times the variance of the jump squared, they converge to zero, then we get a classical not classical, but something that looks similar to classical central limit theorem, with the Kolmogorov and Wasserstein rate of convergence, with Kolmogorov and Wasserstein bounds on the rates of convergence that also go to zero. And I'm not showing the exact rates of convergence here because, they again would not fit on the slide. So thank you for your attention. This was kind of a summary of what I've been working on in the past couple of years based on work that started in my PhD. And this has been published in a couple of works in the past couple of years. So with Vandenin Mitov and Tanya Stadler, we developed this fast inference package for these things, and now with Torkel Erhardsson, we've been developing we've been developing these sort of central limit theorems and rates of convergence for them using Stein's method. So thank you.